那我现在就简单的介绍。那何医师等一下也许要用英文来做 presentation 那不过我现在最怕的就是说，一旦用英文 presentation， 那 student 就 hesitate to ask question， 所以实际上还是请大家可以就尽量的问问题。那何医师是成大医学系毕业那他是念师大附中的师大附中最近有一些新闻那成大医学系毕业之后，他在台大的内科 training， 然后在我们的感染科 training 那接着他就来临床所念硕士班那在硕士班的阶段，他是跟张尚存院长跟王维功教授做 research 那王教授我讲他现在那个王维功教授在我们微生物科他是我小学同学，小学中学高中他来台北念书，他现在在Hawaii State University 过着逍遥的日子。那接着念完之后，他就到 Johns Hopkins 念 PhD 何医师，然后就留在那个地方。那现在他是在 postdoc 留在那边做，然后现在是在找工作的阶段。那 pretty soon 可以看到他可能在几个很有名的大学里面找到他的工作。那我今天请他来谈一下他的 research 以外，可能最后我特别是同学如果有兴趣，谈一下去美国做 research 的 career even 是博士班的学生，你也可以 qualify 完前或者什么时候你要去美国做个2年回来拿那个东西来毕业，我都不反对，好不好？但是最后回来以后，你还是要再做一点东西，好不好？不是说只有拿美国的东西就可以毕业，这是教育部的规定不是我的规定。但是我是很 encourage 同学到国外至少 exposure 2年左右那因为国外的环境毕竟跟台湾是非常在一个非常的不同好不好就这样。好，那我就讲太多，那请大家热烈欢迎何雅琪学姐就是the homecoming 的一个 lecture， 那雅琪来照相。
谢谢老师，非常好的介绍。我前面部分跟最后结尾会用中文讲，希望可以跟大家一起聊。但是中间 science 的部分我可能还是会用英文，不然有很多东西要翻译翻过来翻过去，可能翻得不是很顺。那非常感谢有这个机会跟大家一起分享我在美国所做的事情。我刚开始的时候主要会做virus， 主要是因为在 R1 的时候遇到 SARS， 那时候 SARS 的时候是非常无助彷徨的 R1。但是我们可以根据很多的 guideline 治疗很多的病人，但是我还是不知道这支 virus 到底是什么。那有这个机会，我有2次让到513病房，就是我们的 HIV 病房，跟洪医师一起学着看病人，希望可以更了解这些 virus。对 virus 基本上是非常有兴趣的。可是那时候去美国呢，是因为我发现，即使我对这支 virus 这么有兴趣，我对 SARS 有兴趣，我对 HIV 有兴趣，我其实不知道该怎么样了解它。那所以我就希望可以到美国去接受更 rigorous 的 scientific training。那这也是很好。那我讲一下我的近况是我在2013年PhD 毕业以后，我做了1年的 postdoc 在我们老板的实验室。2015年就升到 research associate， 是一个 non-tenure track 的 faculty。那因为 visa 关系，我没有办法进 tenure track， 所以直到现在都是 instructor。那从大概今年也许5月或6月，我可以升到 assistant professor。那我同时在其他的 institute 在找 job。那这个意思是说呢，我们不一定需要一个标准的 track。把你自己 fit in 进去。通常你去美国，人家就跟你说你要做6年的 PhD， 再做6年的 postdoc， 你再找 job。我跟你说，你的 drive 够强，你不用等，你会有其他的 opportunity 会来找你。那我现在跟大家分享一下我在美国所做的东西。So we know that untreated undetected infection will eventually lead to AIDS. Fortunately, we have this medication called antiretroviral therapy. Basically, can suppress HIV viral load to undetectable levels, and all the patients are living very healthily for many years. However, despite many years of antiretroviral therapy, once the patient stops antiretroviral therapy, the virus will rebound. But why is that? This is because HIV resides in resting memory CD4 T cells as the major barrier to cure. The usual goal of resting CD4 T cells is to become stimulated once there is the antigen coming and stimulating these CD4 T cells into activated CD4 T cells. However, HIV preferentially infects these activated CD4 T cells and even integrate into the host genome and become integrated HIV proviruses, which means that they are now in DNA form. Most of these infected cells will die because of viral cytopathic effect or immune clearance. However, A small proportion of them will return to latent quiescent state with this memory CD4 T cell. They are transcriptionally inactive, and even if you have antiretroviral therapy or cytotoxic T lymphocytes, they cannot see these integrated HIV proviruses. So therefore, these HIV-infected CD4 T cells will just live there for a long, long time. What is even worse, these HIV-infected cells can either undergo homeostatic proliferation or HIV-driven clonal expansion, so that these HIV-infected cells even increase in numbers. Once this antigen comes to our body again, the HIV becomes reactivated, and this is why we have this viral rebound. Therefore, this latent reservoir is the major barrier to cure. This latent reservoir is extremely stable, and if you just keep taking antiretroviral therapy and wait until these infected cells to decay, it takes 73 years to decay to zero. Therefore, given the current 36 million people living with HIV, all HIV-infected individuals have to take antiretroviral therapy. But if you consider the adverse effect, economic burden, and drug resistance, this is not feasible. So a cure is definitely required. However, there are problems. Why can't we cure HIV infection? So first, only 1 in a million CD4 T cells contain this infectious HIV, like this yellow one. So 1 in a million CD4 T cells containing HIV is the standard size of the latent reservoir. The vast majority of HIV-infected cells actually contain defective HIV proviruses. What is even worse, there's no reliable marker which can detect and differentiate cells containing infectious and defective HIV proviruses, so you cannot target them specifically. So the goal of my research is, as a molecular virologist, is to characterize these HIV proviruses in patient cells. I want to understand how these infectious HIV proviruses are reactivated and how these defective HIV proviruses are clinically relevant or not. Furthermore, I hope to identify hosts and viral factors so that we can target them specifically. So the first part is my PhD thesis. It was done a few years ago. So the standard way to measure the size of the latent reservoir, or the way that we can understand how many infectious HIV are there in the patient's body, is called the viral outgrowth assay. Basically, we take patient blood and culture them in limiting dilution, and through some mathematical calculation, we can understand the frequency of infectious HIV in these cells. After 2 weeks of culture, we stimulate them with maximum in vitro T cell activation using this PHA, which is a lectin cross-linker of T cell receptors. We added lots of allogeneic lymphoblasts so that any single virus released in the culture well will be exponentially replicated in this culture well. And after 3 weeks of culture, we can identify whether this well contained an infectious virus using P24 ELISA. So this is the standard method developed by Bob, which is my mentor, 1997. Through this method, this is how we calculated the 1 in a million resting CD4 T cells containing infectious HIV proviruses, and this is the standard size of the latent reservoir. However, if you look at other culture wells, they also have HIV proviruses in them. However, after maximum T cell activation for 3 weeks, nothing comes out. So we think they're mostly defective. Well, yes, they should be defective. So we call them non-induced proviruses. They're generally 100 to 1,000 per million CD4 T cells. When I came in, I'm thinking, are they really defective or not? If there's a patient here telling you that I want to get HIV cure, and you give him some magic strategies to cure them, the patient comes back saying, am I cured? You say, let's check. The standard method is a viral outgrowth assay, and the viral outgrowth assay is 0. But then the patient say, well, you know, we still have all these like HIV DNA in my body. Are they safe? Can I just remove my antiretrovirals? We don't know. So to understand whether these are infectious and whether they're inducible is the main goal of my research. So I developed a single genome amplification method. Basically, we took culture wells, which are negative for viral outgrowth. We collect them and plate them into limiting dilution. We use a full-length PCR to reconstruct the HIV provirus inside these wells. using limiting dilution PCR, we can make sure that each well contains only one single HIV provirus. So when we sequence them, we are not combining virus A and virus B and putting them together into one virus. We want to make sure every single virus that we sequence is from just one viral clone. From that, we found that the majority, 88% of these proviruses are defective. That's what we thought. One third of them contain APOBAC-mediated G2A hypermutations. So APOBAC is a host restriction factor. Basically, we have a virus coming into the cell, they will just make lots of mutations. When you have C to U mutations in the RNA, basically what you get by the end is, look at this GAG start codon. It's changed from ATG to ATA, causing a missense mutation. So the start codon is gone. With all these hypermutations in the tryptophan residue, if any of the G's is mutated to A, this creates an in-frame stop codon. So these 1/3 of proviruses with APOBAC-mediated hypermutations are defective because of these premature stop codons. Around half of them contain large internal deletions, which encompasses these major HIV genomes, such as TAD and REV response element. We know that these large internal deletions arise because of copy choice recombination. Basically, we see that there are short repeats at the junction of these large internal deletions. This is because the reverse transcriptase is very error prone. When it sees one sequence and there is something similar around, it just jumps over and the middle is not amplified. That creates these large internal deletions. With these deletions, these proviruses are defective. We also found a small population of proviruses containing packaging signal deletions. So for HIV, packaging signal is required to package two positive sense RNA together into a virion. And also, there is an important component called major splice donor site in the middle of this packaging signal, because HIV requires lots of splicing to produce essential viral proteins such as TAD and REF. So if your major splice donor is gone, these proviruses are defective. We reconstructed these full-length defective HIV proviruses and tried to see if they infect CD4 T cells. The answer is not. So these parts are also defective. However, we found that up to 12% of these non-induced proviruses contain intact full-length proviral genome, which means that they have an intact promoter, intact packaging signal, and all the viral coding genes. Are they intact or defective? Are they inducible or not inducible? To understand these, we want to check whether they are replication competent, whether they have an intact promoter function, whether they are epigenetically silenced, and we look at their promoter CpG methylation and their integration site. To understand that whether they are replication competent, we put them, this full sequence, into a plasmid using de novo gene synthesis, made them to virus and infect CD4 T cells. If they infect, they're infectious. As shown in this positive control, if you have CD4 T cells, infect them with a wild-type HIV strain, you can see this 2 logs increase of P24 over this culture period. Just the same as our positive control, which are reconstructed induced proviruses from P24 positive wells. Strikingly, what we found is that these intact non-induced proviruses from these culture wells that did not grow anything out, these proviruses are actually replication competent, just like the wild type and their positive controls, showing that they are infectious. Second, we want to understand whether they have an intact promoter function, so we clone the promoter region into a luciferase reporter and transfect those into resting CD4 T cells. So this protocol is modified from Hongqi Yang here when he was in our lab. We're following the same protocol when we did this experiment. This is actually his plasmid. So we cloned this in, and we found that from these patient-derived non-induced proviruses, they have intact promoter function both at resting state and upon stimulation, indicating that their promoter is functional both at resting state and upon activation. The only ones that have bad promoter function are those having APOBAC-mediated hypermutations, because these hypermutations affected these transcription factor binding sites, so their promoter is not as functional as the wild type. Otherwise, these non-induced proviruses have intact promoter functions. We next went on to understand whether they are epigenetically silenced, so then they are not induced. To understand that there are different ways, but the way we try to understand this is called promoter CpG methylation. In eukaryotic cells, if your promoter has lots of CpG islands are not methylated, this indicates that this gene can be transcribed. If your promoter is CpG methylated, it means that your gene is labeled for silencing. Using this method, we took patient resting CD4 T cells, and those cells from P24 negative wells, they're not growing out any virus. We found that these promoters are not CpG methylated, meaning that they have the potential to become activated. We next wanted to understand whether they are integrated into transcriptionally unfavorable regions, such as heterochromatin, like they are all packed up together so that they can be transcribed. We used inverse PCR to understand where they're integrated, and we found that they are integrated into active transcription units. And their activity of these genes are basically active, both in resting cells and in activated cells. So what we thought was maximum T cell activation should reactivate all HIV proviruses. After these, we think that HIV reactivation is stochastic. Stochastic means that for one round, you activate a lot of cells, but another round, you will activate more and more out. They are not maximum, despite maximum T cell activation. Therefore, we took P24 negative culture wells and stimulate them again. We can find that a lot of them can actually be induced again and again, up to 4 times. So the 4-time stimulation was the result published in Jack's Medbauer LabMate like last month. So confirming that this reactivation is stochastic. So what does it mean? It means that we thought that the size of the latent reservoir is 1 in a million CD4 T cells. However, now we think that these intact non-induced proviruses are the true size of the latent reservoir. So if you measure just HIV DNA in patients, you have all these defective proviruses measured. They're not really clinically relevant. If you just use the standard viral algorithm assay, you measure 1 in a million. However, if you use the frequency of intact non-induced proviruses to measure the true size of the latent reservoir, it can be as many as 60-fold more than previously thought. So we show that these intact non-induced proviruses are actually infectious inducible. HIV reactivation is stochastic. We then went on to understand that there are all these defective proviruses there. Are they clinically relevant? I've been showing you they are just defective. So we want to know whether they are defective or not. So this is the postdoc or faculty project that I did, and this will be published hopefully tonight or tomorrow at Cell Host Microbe. So we think that the HIV proviral landscape may be dynamic. And why is that? We want to understand why these defective proviruses are important or not because, first, we know that these latent reservoirs in intact non-induced proviruses, they decay very slowly over time, almost do not decay at all. However, if you look at the frequency of total HIV DNA, we also know that these HIV DNA frequencies do not decay over time. They're just stable. However, recently in 2014, there are 2 Science and 1 Cell papers telling us that HIV can drive clonal expansion of the infected cells. Basically, if you use integration tech to look at all these cells containing infectious HIV, they expand and increase over time. And we think that it doesn't make sense. The total DNA levels stay constant over time, but clonally expanded HIV infected cells increase over time. So what happened? We thought that the HIV proviral landscape is dynamic. So we look at different subsets of HIV proviruses, and we found that the longer a patient is infected with HIV, the fewer proviruses with hypermutations this patient will have, and the more proviruses with large internal deletions the patient will have. So this may be dynamic. Why is it dynamic? We hypothesize that it's cytotoxic T lymphocytes which shape the landscape of HIV proviruses through preferential selection on different subsets of HIV proviruses. We want to understand whether CTO is doing anything to these defective proviruses because we want to know whether these defective proviruses can boost immune responses or they're actually decoys that distract CTO killing of their real targets. To do that, we established an in vitro model and an ex vivo model to understand whether defective proviruses can be transcribed, translated, and recognized by CTLs. For the in vitro model, we reconstructed patient-derived defective proviruses and transfected them into primary activated CD4 T cells. We reconstructed proviruses with packaging signal deletions, hypermutations, large internal deletions, and nonsense mutations. We found that using different methods of detection of HIV RNA inside the cell or in the supernatant, we found that the majority of these defective HIV proviruses can actually be transcribed. Interestingly, we used a primary probe set to look at spliced HIV RNA to understand whether they can produce spliced HIV RNA to make these essential proteins such as TAT and REF. However, these spliced HIV RNA are produced from clones that have spliced donor mutations. What is this? Those with spliced donor defects can produce spliced RNA. I don't understand. Therefore, we cloned the PCR product and we found that HIV proviruses with defective major splice donor sites can overcome these spliced donor defects and splice into canonical acceptors, which means that if their major splice donor is gone, they can just activate something else and splice into their regularly used splice acceptor sites to produce all these essential viral proteins. Interestingly, we found a novel splice donor site that is found from 3 different clones isolated from 3 different individuals. These novel splice sites is actually the GAG start codon over here that encodes the ATG in there. So we're wondering what this final transcript will make, maybe an aberrant protein. We next wanted to understand whether these defective proviruses can be translated. So if they're translated, we can detect them through intracellular GAG staining. So we found that when we transfect primary CD4 T cells with these proviruses, we can see some detectable levels of HIV GAG staining from proviruses with major splice donor defects or with point mutations. Inconsistent with our results from supernatant P24 ELISA, this is another way to detect protein production using supernatant GAG or P24 protein detection in the soup. We found that those with packaging signal deletions or point mutations can still produce detectable levels of HIV GAG. However, those with large internal deletions, even if some of which contain a GAG expressing protein fragment and an intact promoter, they do not produce detectable levels of HIV GAG protein because their TAT and REF producing sites are deleted, so they cannot produce sufficient levels of protein to be detected. To understand whether cells containing defective proviruses can be recognized by cytotoxic T lymphocytes, we collaborated with Brad Jones at George Washington University. So this is what I say about collaboration in the United States. I presented all this talk at George Washington, and Brad came to me. Brad said, "Hey, Yachi, I have something in my incubator. I think we can work on this together." What he had are CD8 T cells that are from specific patients with known HLA types and has been mapped to recognize specific HIV epitope. For example, these epitopes starting with I ends with K is a 9 amino acid epitope that can only be presented by this HLA type, and this presentation can be recognized by a very specific CTL clone. He has all these CTL clones in his incubator. He said, "Yachi, let's work on this together. I want to know whether your defective proviruses can be recognized by my CTLs." I said, "Yes, I want to know too." So we worked together. I basically have 1 bottle of a trizol, 3 vials of my viruses, and all like a big transfection machine. I took this metro from Baltimore to DC just to work in his lab. So we think that if we transfect the CD4 T cells from the same patient with these reconstituted patient provirus, and if anything is presented, and you add the CTL clone in, if the CTL is recognizing anything that is specifically presented by my plasmid, we can detect CTL recognition through CD107A flow cytometry. So if it's recognized and it's activated, then we can detect this. So we found that in our proviruses with packaging signal deletions, when we transfect the provirus in, these proviruses have matched CTL epitopes. So when we put CTL in there, we found that they are recognized by CTLs as shown by CTL recognition and CD107A expression. Very surprisingly, we have proviruses with APOBAC-mediated hypermutations. Basically, these clones have matched CTL epitopes. However, there are lots of premature stop codons before them because they are hypermutated. However, when you transfect these proviruses into the CD4 T cell and add the CTL clone in, we see significant levels of CTL recognition from 2 GAG-specific CTLs and 1 NAP-specific CTLs, which means that 1, something is specifically recognized by cytotoxic T lymphocytes. 2, they are from proviruses that have premature stop codons before their recognition site. So this is very interesting. And third, we transfected these proviruses with matched CTL epitopes but large internal deletions elsewhere in the genome. When we put this in, we found there's no CTL recognition, so indicating that these proviruses with large internal deletion are probably not recognized by CTLs, and probably they do not encounter any selective immune selection pressure. So to confirm whether these happens ex vivo in patient cells, not just in plasmids, we first took patient resting CD4 T cells from patients under antiretroviral therapy. We activate them. So after we activate it in the presence of ART, we'll see lots of viral RNA transcription, but they don't infect each other. We should be able to see HIV RNA, which are hypermutated or not hypermutated. And this is true. We found that both in resting CD4 T cells and activated CD4 T cells, there are a significant proportion of these cell-associated HIV RNA that are actually containing APOBAC-mediated hypermutations, indicating that these cells containing hypermutated proviruses can actually be transcribed from patient cells. We next want to understand whether these alternative splicing patterns happen in patient cells. So we did the same thing. We took resting CD4 T cells and activated them in the presence of ART. We found that there are interesting clones having large internal deletions encompassing this major splice donor site, and they can activate an alternative splice donor site to splice into a canonical splice acceptor site, which means that even when this major splice donor is gone, they can still splice and make these essential viral proteins. We next want to understand whether the CTL recognition of hypermutated proviruses happen in patient cells ex vivo. So we took patient resting CD4 T cells, activate them so they will express HIV RNA. We expect to see HIV RNA that encodes wild-type CTL epitope or escaped CTL epitope. And also, they can be wild-type or containing escaped epitopes with premature stop codons before them. So if this happens, we're expecting to see all these 4 populations. And this is what we see. We activated these CD4 T cells and we added CMV-specific CTLs, which means that these cytotoxic lymphocytes are stimulated by cytomegalovirus, which is not HIV, and they should not kill HIV-infected cells. And adding these in, we still see a lot of these APOBAC-mediated hypermutated HIV RNA in these populations, indicating that, yes, we do see all these 4 populations, and hypermutated HIV RNA can be transcribed. But when we added HIV-specific CTLs, which means that we took patient PBMC and simulated them with GAP peptides, so then we will expand these clones with HIV-specific CTLs, we found that these hypermutated HIV RNA just decreased and some are just gone, indicating that HIV proviruses making hypermutated RNA, these cells can be recognized by CTL. So this is very interesting. We propose possible mechanisms of these alternative HIV protein presentation. These proviruses having hypermutations can be transcribed and translated through alternate reading frames, alternate start codons, ribosomal scanning, and defective ribosomal products, which means that these proviruses can be transcribed, not at a very high level. They can be translated. However, they can be presented better because they are just considered as junks in the cell. So they are presented faster and presented better. The reason why they're presented better is because HIV encodes a protein called NAF. NAF will downregulate MHC1, so to hide the presentation of their viral proteins to CTLs. However, our defective proviruses, they do not have NAF. They're either hypermutated or they're deleted. So they cannot downregulate MHC1, so all these epitopes will be presented to CTLs. So they are not necessarily transcribed better, but they're recognized by CTLs better, and that creates a CTL selection pressure. In summary, we thought that HIV-specific CTLs should only recognize cells containing infectious HIV. However, we now found that defective proviruses containing major splice donor site defects can be transcribed, translated, and recognized by CTL. Proviruses containing hypermutations can also be transcribed, translated, and recognized by CTLs. Those proviruses containing large internal deletions can be transcribed, but they are not recognized by CTLs, and over time they will just increase because of the lack of immune selection pressure. We are still trying to understand whether they are decoys or they are actually a good vaccine candidate. So the third part I'm going to talk briefly about is what I'm going to do in the future. Because by now, I talked to my husband that I'm working on HIV cure research, and he will always emphasize back that I'm actually working on HIV no cure research, because I tell you that you think this is this big, it's actually even more. You think that they're defective and you don't need to care. Actually, you do need to care. So this is just very sad for the field. I still want to find a way to improve HIV cure. So what can I do? As the molecular virologist, I want to identify a target, either host or viral factors, so then we can identify and kill these cells. As I talked briefly, we know that HIV may drive clonal expansion of infected cells through integration of cancer-related genes. In these science papers published previously, they found that HIV may integrate into cancer-related genes. For example, this is MKL2, and in these cancer-related genes, see, HIV integrate into this small proportion of these introns, and they're all in the same direction, and they repeatedly integrate into this small spot. And this is confirmed in 2 different studies from 6 different patients and 2 very distinct study groups. So how could this happen? Why do HIV preferentially integrate, or why are they enriched in these cells integrated into these cancer-related genes? We want to understand whether these HIV-infected cells, whether HIV integration into these cancer-related genes will facilitate the proliferation of these infected cells. However, the easiest way to do that is to take the cells and do a transcriptome analysis to see what's being transcribed, what is not. However, there's no reliable marker which can identify HIV latently infected cells. And if you just take patient cells and RNA-seq all of them, you were just doing RNA-seq on the bulk majority of either uninfected cells or cells containing defective HIV proviruses. So, and we developed a method called RNA sort-seq. So this is my R21, the first grant I got in 2014. So we developed this method. Basically, we'll first reactivate patient resting CD4 T cells, and I use this fluorescent in situ hybridization using 96 probes at 5' HIV, 96 probes at 3' HIV. So then, if these cells contain HIV RNA, I can sort them out and just sequence these HIV-infected cells. So this is what I did. We found that our 5' and 3' HIV labeling is very specific. As you can see that they co-localize in the cytoplasmic pattern. We can therefore sort HIV-infected cells and compare them with HIV-uninfected cells for transcriptome analysis. While I'm still working with my bioinformatician to understand what's being transcribed and what's enriched, we found that there are very interesting sequences in these cells. We can find HIV integration into a host intron. We can find the exact junction of the host genome and the HIV provirus, either the same or conversion direction. And some of these genes that they integrated are actually cancer-relating genes. What is even more interesting, we also found host exons that it should be spliced into another host exon, but it's actually spliced into HIV, into a canonical known acceptor. And we don't know what these aberrant splice product work, but we think it's very interesting. For example, this part is a TET encoding region, and we don't know what this protein means, and it may activate some of the domains. And the integration sites are actually all in cancer-related genes, and some are even reported in the previous 2 science papers, indicating that there is something going on, not just happening by chance. So in conclusion, as a molecular virologist, I understood how far the cure would be, because I now know that these non-induced proviruses are reactivated through stochastic reactivation. And we know that defective HIV proviruses may actually be a decoy, and this may alter the CTL killing. What I'm working on right now is to use a transcriptional analysis to understand what these HIV infected cells look like. What I'm actually developing is a CRISPR-ready epigenetically characterized cell line model to allow high throughput screening for HIV silencing agent. So the future direction I'm going to do in the future, the first part is that I've developed this method to look at HIV full-length sequencing, and we call this HIV DNA landscape. This is considered as my collaboration with my mentor, Bob, so I'm going to move away from that because in the United States, you seek independence. For independence, I have different directions. First, I'm going to map HIV RNA landscape, because this is the first method that we can identify HIV integration site and their replication competence in one single method. And second, we want to understand how we can enhance CTL selection pressure through hypermutated HIV proviruses. Third, we want to interrupt HIV clonal expansion through understanding of their expansion dynamics. We're currently working with Steve Deeks. We just submitted the grant Friday before I flew out. So they have a clinical trial that fit what we need, so we're going to work on this together. And fourth, I'm going to identify HIV silencing agents. This was previously funded, and I've submitted this grant with David Knight at Harvard. So we can, because when I gave this talk at Harvard, and David Knight, who was the director of the department in BIB and also the chief editor of Field Virology, came to me. He said that he's been working on HSV silencing for a long, long time, but now I'm working on HIV silencing. He hopes that we can work together. So we work together because he's done lots of high throughput sequencing, and I've done lots of HIV work. So we put this together into a grant that was submitted last December, so then we can work together to find an HIV silencing agent. So I have a few minutes to tell you about why I want to stay in the United States. Tenure track. When you hear the word tenure track, you think that, oh, okay, now you are like attending a 5th year, you get this PhD degree, you get to assistant professor, you get 3 more papers, you get associate professor. 10 years later, you go to professor. No, this is not the tenure track in the United States. Tenure track is brutal in the United States. It means that you really need to be the one that when everyone look at you, you are the hero in your field. And this track is very difficult and can sometimes be frustrated. You know all of them. Sorry, this is my favorite director. He is the director of Berlin Philharmonic. So it's not easy. You have lots of times you're lonely. No one knows you and no one knows what you're doing. You can't even persuade your family to pursue a track that no one knows what you're working on. But if this is your passion for your life, there is a drive. So I'll show you. So what's your passion? You think that, okay, going to ophthalmology is your passion. That is great. But this has to come from you, not from your parents or your classmates, or because that makes money. You love it as your lifelong career. Go ahead. I don't care which subspecialty you take, but it has to be your passion. You come here and do a PhD, I hope this is also your passion. Not that, oh, everyone is also taking it, so I just have to take it too. Please don't. It's going to make your life miserable. Second, are you ready and are you afraid of taking challenges and risks? Here, well, you have some risk. If you don't have a paper, you probably won't graduate. And in the United States, it's even worse. If I don't get a grant, I'm out. Like I have assistant professor, associate professor, full professor, whatever title you have, you don't have a grant, you're out. And are you afraid of that? Yeah, I have mortgage to pay. But do you want to do it? Yes, I want to do it. Because challenges and excitements are the two sides of the same thing. How much do you want it? I want it. There is a drive that just say, I just want to do this. Going to the lab, do research, pipetting at the bench is the thing that I want to do most in my life. Really, I tell you, this is really. I want to do it, and I don't care how much challenge that is. Challenge means excitement. Don't be afraid. And third, the real question is, are you ready to take these challenges? You can take challenges, but only if you're ready. All of these training, PhDs, postdocs, residency, those are the process to get you prepared for something you want to do for your life. After residency, you're very comfortable. You can take care of all these critically ill patients, no problem. After PhD, you are not so sure. But it depends on how much you want to make yourself ready. If you only want to get a degree, 5 years later, you'll get a degree or whatever you do. But if you want to really take science as your lifelong career, you have to make yourself ready, not your mentor. You have to do it. Last question. Should I get this PhD degree? Should I get to the United States? When should I get to the United States? It doesn't matter. You don't need to take my track. Find what you want. Find what you love. You have family, you have career, have patients there. You have dream in the United States. Could be bad or could be good. It doesn't matter. Follow your passion. You are either 30 or 40 at this stage. You have endless energy and possibilities. Follow your passion at this very moment, and that will lead you to what you really want. Okay, so thank you. I'd like to take questions.
最后那张 slide 也是讲了以后再加进去的。OK， 好，那前面有没有大家要讨论的问题？就这个，你这个也是做 HR 你先问来。
雅琪，谢谢你非常精彩的演讲。两个问题啊，第一个问题简直就回归到你的题目 ，How far away？ 你觉得我们大概还有多久我们可以走到所谓的 cure？ 那第二个问题就是我们很有兴趣，就是说你最后讲到所谓的epigenetic silence。然后这个的话，问题是epigenetic silence 你必须要分HIV gene specific 的 silencing，overall 的 silencing， 我想大概赛更大，效果很提升。不知道你对这两件事有没有什么看法？
Wonderful questions. 有一个 American Institute called American Research Foundation of AIDS， 他们就从那个什么Leonardo DiCaprio 还有那些 celebrity 捐了非常非常多的钱，每年就会给很多个 million grant 来给大家做 HIV research。他们的第一页首页就是 Countdown to a Cure 2020 3 years later. Do you believe it？ 答案是没有啦 ，grant 可以写，实验可以做 ，cure 没有那么近。但是 cure 在哪里呢？你就会问我说 ，OK， 那我现在问题是 ，OK， 你现在给我$1 billion， 我可以做任何我想做的事情，到底怎样会 cure？ 现在有几个比较 promising 的 direction， 但是很抱歉不是我做的，但是我要跟你说那是别人做，但是我很羡慕他们。第一个叫做 Lewis Picker CMV based HIV vaccine， 他们发现 CMV 有一种 specific 的 presentation 方法，他们用 HLA-E 去 present 这些 SIV 或者是 HIV 的 antigen， 那已经在做 clinical trial， 他们在 SIV 看到效果非常的好，不管你的是什么样的 MHC 态，它可以当成一个很好的 vaccine， 这是一个很好的 cure。那第二个 cure 呢？ Maybe T cell therapy， 我不确定。就是我们现在的 goal 是，以前我们希望可以有 sterilizing cure， 就是你要把每一只 virus 都杀死。但是至少我发了 paper 以后，我跟你说，总之是杀不完的，所以就不行了，就 shock and kill 到这里就死掉了，就不能再做了。所以呢，你要做 functional cure， 就是你如果可以 boost immune response，no shock just kill。你如果 immune response 就够强的话是可以的。第二个问题叫做 epigenetic silence 要怎么样 specific？ 对，很抱歉，我有一个那个 grant。 我来之前 letter of intent 过了，刚好2个礼拜后要交，他就是说你这个怎么可能 specific？ 我也说对啊，我不知道。所以呢，这跟 shock and kill 是一体的两片。shock and kill 就是说呢，在过去，从2012年开始有非常非常多的 paper 呢，在做 clinical trial， 如果我们用HIV latency reversing agent， 你可以把这个 HIV 从 latency 里面吹出来，再把它杀掉的话，这样也许可以 cure。那些药叫做 HDAC inhibitor， 也是一种 epigenetic silencing，no specificity against HIV。那些做了一大堆 clinical trial。 那我们只是想说呢，我们希望可以找到一个可以 preferentially inhibit HIV 的东西。那 NIH 去年12月就是发了这个 grant， 就是我跟 David Knight 一起写的这个 grant 呢，他们想要找，那我想说好啦， NIH 如果想找就来找吧，找不到就算了。就是你至少有一个人要有一个最 rigorous 的方法去找。那没有我就跟你说没有，那试试看，不知道可能没有。
OK 好，来那个蔡宏强。
真的完全的随机，还是说是观察到每一次会有
？Same question， 我根本不相信是 stochastic activation， 因为你后面实际上你看到不同的 chrome， 不同 mechanism， 它的 proviruses 跟那个就不是 stochastic， 对不对
Mathematically definition stochastic 就是1次、2次、3次、4次出来都是一样，就是 stochastic， 但是没有说为什么 stochastic。这问谁呢？你这个问题我在 UCLA 遇到一样的问题，那是我就问 Alex Hoffman， 他是专门做 stochastic， 他是这样说理论上是，你真的要去证实呢，你要把细胞一个 sort 进那个 single cell， 然后让他们 divide， 让它 stimulate 以后左边跟右边这个他们的 reactivation 是不是一致的？有 MIT 有人在做，可是不是我的 collaborator， 我没有办法找他做。真的要问的话是要那样子问，所以它是不是 stochastic？ 它不一定是 stochastic。以 mathematically 这个现象是 stochastic，biologically 不一定是。那就是很多 mechanism 比如说，如果我的 RNA fish 可以找到什么样的方法说啊，其实就是一个 transcription factor 有了它就 reactivate， 没有了就没有，那我就可以把 stochastic 变成 deterministic， 那我就可以跟你说会有一个方法可以把它们全部都 reactivate 出来。目前不知道 ，mathematically 是 stochastic，biologically 可能不是，但是不知道为什么。
那另外其实还有来黄彦琴来。
谢谢。我有几个小问题啦。第一个就是14年那个 Brazil 他们看到那个 baby 啊，Mississippi 的那个 baby， 他可以有一段时间好像3号好像是 function cure。我在想那个中间过程还有发生什么事，就是以你的这套的理论来讲，套不套用在这个人身上，就是这个小朋友身上。那当然我们现在 HIV 的治疗现在是可以长期的而且有效的抑制。我不太晓得这个，我想您可能这边的对象大概都是已经受过长期治疗的对象。我想这一套理论是一个没有被治疗的 ，specifically 是那个所谓的elite control 的。不太晓得这一套他们在这里看到的，像刚刚所谓的 non-induced 的一些 non-inducible 的这个 cell 的这种 proliferation， 在这样的 non-inducible， 就是所谓的elite control 这样子对不对？
我得回答第二个问题，这个问题非常好。这个问题呢，在我来之前还在做，我们拿了一个Gilead grant， 就是2.5个 million， 我跟我老板跟 Tom Cruise 3个人分的，我们做就是这件事情。就没有人知道 elite control。elite control 的意思就是他们不需要吃任何的药，但是他们的就是 viral load 就可以用自己的 CTO 把它 control 得很好。但是我们不知道为什么。那洪医师的问题说，这些 elite controller 这种 activation 是不是还有同样的 pattern？ 答案是没有人有办法做 full length sequencing， 因为他们的 virus 太少了。那我在来之前，我们跟 Steve Dix 刚好就是拿了那些血，就是他们有 elite controller， 而且他们是 antiretroviral therapy treated， 所以我们可以不会看到它的 ongoing replication。可是同样在 ART suppression 之下，它的 sequence 长什么样子？我们看到绝大部分都是 defective， 但是还是有找到一小部分的 intact。那不知道他们是没有被 induced， 还是他们就是刚好就有什么样的 proliferation 在那里。所以答案是 elite control 我们不知道，但是这是 ongoing 正在做的东西，而且看起来蛮有趣的。那第二个问题是 Debbie Prasad 所做的 Mississippi Baby， 历史上有几个有名的例子是 HIV cure， 比如说 Berlin patient 他接受了一个 CCR5 delta 32的 bone marrow transplantation 以后他就 cure 了，再也找不到 virus， 这是一个人。第二个人叫做 Mississippi Baby， 在 New England Journal 上发现的有一个小朋友呢，他在小时候出生以后感染，结果 lost follow up 回来以后，都没有 HIV， 都没有 rebound， 他就觉得哇，这一定是 cure。结果过了几个月以后，很不幸他就跑出来了。所以我们认为呢，这个过程只是因为它的 reservoir size 非常的小。假设我今天得了 HIV 呢，今年的 Croy Gintana 讲的事情是 you get what you start with。如果我刚开始的 reservoir size 很小，比如说我是一个 baby， 我出生第一天一有 HIV， 我马上吃了 antiretroviral therapy， 我的 size 就会很小。如果我过了5年呢，我才开始吃 ART 呢，我的 reservoir size 就会非常的大。那在这个 Mississippi Baby， 我们现在认为呢，是因为它的 size 非常小，我们目前这些。假设我今天得了 flu， 明天 measles， 后天又在接受一种奇怪的 antigen， 我每天会有 by chance 会有一些的 virus 跑出来，就是一点点。那 那刚好这个 Mississippi Baby 它到跑出来by chance的时间非常的长，让大家认为有一个 functional cure 或者是 remission 的 period， 但是实际上它并没有达到真正 sterilizing cure， 然后我也不认为它真的有 immune 的 control， 所以它不算是真正的 immune control， 它只是 by chance 还没有到而已。
好来，3分钟。
跟杨医师都在做 HPV， 我们在做 HPV 的 Q， 所以基本上，不过我们有一个想法是说，假如说 for invention to cure， 这样 you have to use immunotherapy， 这是我们的想法。对，那所以我们在一个有趣的问题，就是我对你的 RNA sourcing 有点兴趣，因为我们现在也在试着要去确认到底一个肝脏里面有多少个 hepatocytes 是一个 HPV infected 或者是 integrated 的，所以你的那个 sourcing 它的 scientific 跟 specific 可以跟我们分享一下。
RNA 蛮好的，因为一个细胞会有非常多 copy 的 RNA。如果你的 HBV 有 express RNA，那就没有问题，我们可以商量那个 protocol 出来。但是如果是 DNA 就非常的难。DNA 的 fish 目前做的最好的应该是 telomere， 就是 Carol Greider 做的，他们在做 telomere 的 DNA fish， 因为 telomere 有非常非常多的 copy。但是否则呢，假设一颗细胞里面只有一个 copy 的 HIV 或者是一个 copy 的 HBV， 然后你要把那些 fluorophore 全部都粘上去，那可能会有一点困难。所以主要决定点是你有没有 RNA transcription， 有就可以做。那来做，现在来做。
问题你们是不是在肝里面？
对，它就是要
不一样嘛，人家这里是可以抽血，这两个就不太一样。
因为我们可以估计它的 size， 比如说当我们要去做这个治疗之前，我们必须可以估计它的 size 到底有多少它都在被感染。
我们就是 Hopkins 有一个人叫 Ashwin Balagapal， 他是 ID 的 physician， 他拿了那个 liver transplant 的 explant， 整颗 liver 拿出去呢去做 laser microdissection。但是即使是这样子做呢，他的那个 RNA preservation 都还是很不好，就 RNA quality 不够好，所以没有办法做出 RNA-seq。但是因为就我这样嘛，就是这样老师讲，我就是血就抽出来就做了，所以我的 RNA quality 非常好。那肝脏拿过来的 transfer 的，这我就不知道了，比较难一点。
那最后这样啊，我对学生讲一下，就是要追求自己的 passion 啊，尤其我们所的学生啊，你临床工作啊，郑亚中应该知道嘛，你有 passion 嘛，哈，对不对？就是要 passion。那 passion 一个目标，我想这个 passion 绝对是来自于对 science 跟还有对病人的 commitment， 这个绝对不是。如果说你的 passion 是来那个 glory 啊，或者是 fame and fortune， 这个绝对不是一个好的行业啊。大家最近的例子已经知道了嘛，对不对啊？他们拿的 grant 可能是我的上千倍啊，结果那搞成这个样子，所以这个请大家要注意，这个 passion 是很重要，那个 passion。然后另外一个就是要 take risk， 好不好？因为我们不知道，就是说雅琪出去，你会知道你能够混到今天吗？对不对哈？他也许当初你会，比如说像陈佩荣医师也是这样，他念硕士班第一届的时候，我本来是要叫他直升博士班，他说不干，他要去 Johns Hopkins 念 PhD， 好不好？那刚才讲说在美国都说 for independence， 在台湾是说 for inbreeding， 好不好？所以你要跟着老师扒着他大腿，你就可以永续经营但是有时候经营就会出问题老师跟学生都会出问题，所以请大家要注意这个事情就是真的找到有兴趣的事情，无论你是在台湾做或在美国做你去美国学了再回来台湾或者什么都可以。那像王维公老师是去了美国回来台湾，还有台湾又去美国所以这个就是你要追求你自己心里在想的事情这个是最后特别要强调的地方。然后何医师这个接着在感染科还有演讲包括在14B 在14B 还有演讲，所以如果还有进一步有兴趣的特别比较专题的问题可以在那边再去听他的演讲那我们时间的关系，我们2点还有另外一位 speaker， 我们就最后再热烈谢谢何医师
谢谢。